Alrighty, crew. Good afternoon. How are we all going? Can everyone hear me okay? See me okay? Hopefully, we're all doing well for this wondrous week 10. How quickly time flies. It is rushing by at a million miles an hour, guys. It's unreal. Oh dear. So, this is our second last week of content. And these next 2 weeks are going to be, how can I describe it? Probably a refreshing change of pace would be the best way that I could describe it. At the moment, guys, from where you are sitting right now, the hardest and most grueling part of the course is done. These next 2 weeks will be a lot more familiar to you guys, as in a lot of the things that we're going to be talking about are things that you've sort of learnt about before, things that you've covered in other more introductory courses, perhaps. So it should be good. Now, a bit of a heads up. So your second assignment is due tomorrow. Tomorrow. I saw tomorrow over here, and that was just what came into my mind. Next week, so it'll be Thursday next week. I believe the link to upload that through Turnitin is under week 11. Basically, I've set a deadline on that for 1:40 p.m. next Thursday. My advice would be to do it sooner rather than later, just in case of any IT or tech issues. Okay. So yeah, make sure you hook into that. Then, so next week is your second take-home assignment. Then, week 12, our final week, will be a big revision week, and you will do your third and final course quiz, which I'm going to have a chat to you guys about that in the shoot this afternoon. And then it's exam time, which I've pretty much finished your exam writing. So what I'm going to do is during the tute workshop this afternoon, I'm going to give you guys a bit of a rundown on what you're in for the third course quiz. We'll have a bit of a chat about the second course quiz. We'll have a bit of a chat about your assignment and, of course, your end of trimester exam. But on that note, guys, let us get started. So what are we doing here? What are we doing this week? Essentially, what we're going to do is continue on a little bit further on our discussions on lipids, and we're going to be looking more specifically at biological membranes. So our learning objectives just from the get-go are just a stark difference compared to what they were for the rest of the trimester. What we're going to be doing is a very, very brief recap on lipids that we did last week. And then we're going to be looking and discussing the fluid mosaic model, and we're going to be looking at some of the factors that can affect membrane fluidity. We're going to be looking at how lipids can move in and around the lipid bilayer. Then we're going to be discussing some membranous proteins. So as I'm sure you all remember, all the way back in week 3, I basically said that within this course, we're going to be covering 3 major sort of types of protein. We're going to be looking at fibrous proteins, globular proteins, and then I said, and way, way, way down at the end of the road, right at the end of the course, we're going to do membranous proteins. And, well, that's where we are now. So we're going to be looking at membranous proteins, very briefly, just in terms of the structure and function, and then we're going to have a bit of a look-see at some hydropathy plots. So yeah, nice and straightforward, which is good. Wrong button. There we go. So let's look now at our membrane components, looking at lipids and cholesterol, okay? What are we looking at here in terms of the plasma membrane, okay? The plasma membrane is incredibly important because it is able to control the movement of various substances into or out of the cell, okay? It's able to sort of contain and separate substances and that's very important because a lot of these intracellular chemical reactions are incredibly sensitive, so we need to be able to control what moves in and what moves out, or what doesn't move in and doesn't move out. A perfect example would be something like action potentials, and how that relies so heavily on the electrochemical gradient on sodium and potassium. They also contain a lot of these receptors, okay, for cell-cell communication, to be able to, turn signaling pathways on, to be able to turn them off, okay, to control a lot of these important cell-cell communication, which, we absolutely need to survive. And it will contain a fuzzy coat of glycoproteins and glycolipids, which, again, as I've said, are incredibly important for cell attachment, cell recognition, and cell communication. Now, what I'm essentially talking about here is that, yes, we can try and control movement of substances into and out of the cell via the cell membrane, but sometimes our control over that is a bit limited, and it depends on the chemical properties of that substance that is trying to move through. So, for instance, obviously the cell membrane is made of a lipid bilayer, so any sort of fat soluble or any hydrophobic substances can pass through this lipid bilayer easier than, say, a water soluble substance, which again we'll talk about secondary messenger systems very, very briefly in a moment. Now, this is just a nice little recap of what we were looking at and what we were doing last week. So when we're looking at our cell membranes, these are the main constituents. We've got cholesterol, we've got our phospholipids, we've got sphingolipids, we've got carbohydrates, and we've got proteins. And different combination of these things will form different constituents of the cell membrane, whether it be the lipid bilayer or looking at our glycolipids and glycoproteins. Of course, this lovely bad boy down here is cholesterol, okay, which we're going to talk about a whole lot more, especially when it comes to the cell membrane, that the membranous fluidity. Now, remembering this is our sphingosine here, so this is our sphingosine backbone, and of course our glycerophospholipids, so we've got our glycerol backbone there. Okay, now, I'm sure that we are all aware of this, but regardless, I do want to just quickly recap that when we are looking at the plasma membrane, it is made up of a hydrophilic, so water-loving polar head, as well as these two hydrophobic fatty acid tails. And what's also important to note is that these aren't rigid, okay? That would make it very, very difficult for us to sort of exist. These aren't sort of rock hard and rigid. These are very fluidic. But the big thing is that these are still lipids. We do see or the disruption of these hydrophobic interactions between fatty acid tails with things like a change in the saturation of the fatty acid or the length of the carbons. And indeed, these are also sensitive to temperature. So when we look at that, we look at the the lipid bilayer and these hydrophilic head. Big thing here, and again, I've said this every single week from week 1 to right now, it is looking at these non-covalent interactions, okay? It is looking at these secondary attractive and repulsive forces. Okay, so what are we exactly talking about there? Well, when we're looking down here at our lipid bilayer, there's the attractive forces that we are seeing an attractive force between these hydrophilic heads and the sort of between these hydrophilic heads to each other, tails, and the aqueous environment of that interstitial fluid or extracellular fluid. Because it's hydrophobic, it's wanting to get away from that and sort of move in. And we're also going to have more secondary attractive forces between these fatty acid tails. Now, in our wonderful world of biochemistry and looking at lovely textbook sort of drawings and stuff like that, It makes it look nice and straightforward and nice and simple. Unfortunately, in the real world, it's not as straightforward as this. Membranes can actually be incredibly complicated. Obviously, with the whole, surface, cell receptors and looking at glycoproteins and glycolipids and, looking at your different integral and peripheral proteins and all that sort of stuff, even just removing that sort of stuff, the raw composition of a lipid bilayer is actually quite complicated because it all comes down to structure equals function. And many of these different lipid bilayers have a different function, which means it only makes sense that they will have a different structure. And that's what I'm kind of saying here. The fundamental structure and function of all cell membranes depends on the different lipid compositions. Now, what is it that I mean by that? Well, if we have a look here, not only are we looking at a lipid bilayer in terms of the cell membrane, but what's also important to sort of keep in mind here is that the definition of an organelle is an intracellular compartment that has a lipid bilayer. So, and because all of these different organelles have different functions, they will actually have a different composition of various lipids. So a couple of examples here, like cardiolipin, it's a major component of the inner mitochondrial membrane, but not of the plasma membrane. And what we're seeing here is cholesterol is very, very prominent in the plasma membrane, but we barely see any of it in mitochondrial membranes. So different membranes within different organelles within different cells will be comprised of different lipids because they are performing different functions. Now, would I expect you to know or have memorized these various composition of lipids in different organelles? Absolutely not. No. That's a bit ridiculous. What I would expect, a lipid bilayer within the human body is made exactly the same and exactly like this. No, there's obviously a heap of variation between these lipids. Now, another thing too I want to just add on to this is not only do we have a large amount of variation between the lipid composition of various membranes, so looking at, again, these different organelles or different cell types, we also have a difference in the lipid composition between different layers of the same lipid bilayer. So, for instance, the outermost leaf, if we're looking here, this outermost leaf or this outermost layer of the lipid bilayer, it could be a different lipid composition than if we compare that to the innermost layer. So there's a whole heap of different variants here because it all comes down to what the overall function of that cell is. But talking about these lipid, the lipid bilayer, and looking at how the membrane sort of responds, the best way to sort of think of it is looking at this fluid mosaic model. Now, essentially, what this is describing is the more physical nature of the lipid bilayer, and a really good description of it, which I really, really liked. Is the membrane it's in a constant state of motion. It's not rigid. It's not just like a block, and it's like a sea. It's like a phospholipid sea, and it's just got different proteins floating around inside of it. So a really good way to sort of process it in your mind is like, imagine you're at Wet n Wild, or you're at a water park, and they've got the big sort of big wave pool sort of thing, and it's absolutely packed full of people that are sort of hanging onto like floaties, and they're just floating along on the waves there. That's pretty much what we are seeing here. Okay, it's basically a phospholipid sea with things floating around and moving around in it. Okay, now, speaking of motion within the lipid bilayer, there are a couple of things that we need to discuss here. The first one, membranes aren't static, and that's incredibly important. Membranes are in a constant state of motion. So what is happening here is that these layers are going to move around, especially by lateral diffusion. And the speed of that will be determined essentially by the degree of unsaturation. So the more unsaturated a fatty acid is, the quicker it will be able to move. And again, this has to do with the amount of, secondary attractive forces that are occurring between these fatty acid tails. Now, there are two main types of movement that we can see here. The first one, as I just said before, is lateral diffusion. Now, what do I mean by lateral diffusion? That is essentially the ability for these lipids to move side to side. Pretty easy, okay? That's not too bad. And this has been observed experimentally by basically putting fluorescent dye from two different cells and getting them to sort of merge together. And you can actually see that that dye sort of fusing together as it's moving around. What is much harder, however, is transverse diffusion. Now, this is where we flip-flop, which is just a great name. So when we flip-flop, that's basically when a lipid that, say, is pointing on the outside of the cell will flip and point into the cell. So this is like what we're seeing here with flip-flop. So it's up the top, and then it flips down. This is way, 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 way harder because it is very energetically unfavorable. Also, I just realized my face was like totally in the way when I was explaining that. Sorry, this is what I meant here. So facing sort of the outermost layer, and then it will flip and face the innermost layer. Now again, this is very, very slow because it is so energetically unfavorable. But how we can speed this up is by the use of enzymes, okay? Enzymes come to the rescue once again. And we have a wonderful little enzyme called flipase. And what does flipase do, I hear you ask? It is a great question. It flips the lipid. it helps facilitate that flip-flop, okay? And by flip-flop, I mean turn it upside down. So, big thing to take away from this slide is the lipids within our lipid bilayer are in a constant state of motion. They're constantly moving around. These lipids can move side to side with relative ease and quite quickly. However, flip-flopping, so going from outside to inside or inside to outside, is far more difficult because it's so energetically unstable. Or unfavorable, I should say, rather. However, we can rely upon the use of an enzyme to help speed that up. Okay, now, let us talk now on the degree of fluidity. Because as I've said to you guys last week, when you take a lipid, or these fats, and we look at some of them which are a liquid at room temperature, and some of them are a solid at room temperature. And the main factors that control that are the degree of saturation. So is it completely saturated? Is it monounsaturated? Is it polyunsaturated? And the second thing is the length of the carbon chain. The longer that carbon chain is, the more, the higher that melting point will be. Whereas the more unsaturated this fat is, the lower that melting point will be. Because it's that kink in the chain will disrupt those non-covalent interactions between those fatty acids. Okay, that's all well and good. Just because these fatty acids and these glycerophospholipids are part of our lipid bilayer, they're not exempt to that rule. This rule still applies to our membranes. And what we see here is, like, we have our paracrystalline state, which is very, very, very... this basically doesn't happen in humans. It's disastrous. But what we can see here is, like, very sort of saturated fatty acid tails, very close together. There's not a lot of movement there. It's very rigid. But as we say, heat things up or change the composition of these fatty acid tails to be, say, shorter in length or be more unsaturated, then what we see here is a lot more... disruption, I guess we would say, and they're much more fluidic, okay, in terms of their ability to move around. Now, what we see here in this lovely sigmoidal graph is this sort of transition between a more fluid-like membrane fluidity compared to a more rigid and solid fluidity. So again, this is just another schematic to help explain this here. Now, this transition, as I've just explained, is the length of the fatty acid tails and also comes down to that degree of saturation. So the more interactions that we have between these lipids, then the more rigid that membrane will be, because it's obviously going to take more energy to disrupt those interactions and allow them to move around more freely. Okay, so that's all well and good. Okay, so we've got our transition temperature here, where we go sort of from solid-like to fluid-like. But the thing that's a bit of a problem here is this. This is a really sharp curve. Okay, this is really steep, and there is a very little temperature gap. From when the membrane will go from more rigid and solid-like to more fluidic. Now, if we suddenly go, look, we want our cells to be more rigid, we can't just sort of completely replace the entire lipid bilayer. You know, you can't just chop things off and attach it here and there. We need an easier way to help control this transitional temperature. So from when the lipid bilayer is going to be more fluidic or more solid, because this is a really, really steep curve. So how do we do that? Well, this is where cholesterol comes into play. Now, what cholesterol essentially helps us do is, instead of having this really sharp curve, this really sharp sigmoidal curve here, it essentially flattens it out. Now, this can be tricky, because if you look at cholesterol on lipid bilayers just at its face value, it's doing opposites. It's doing the opposite thing. It's both making the membrane more fluidic and less fluidic at the exact same time. Okay, so it's a bit of a like, wait, what? How can it do that? How can it make it both more fluidic and less fluidic at the same time? That doesn't make any sense. When you say it like that, it doesn't, but allow me to explain. What it does is it flattens this line. So let's say, look over here when it's at a higher temperature. So at a high temperature, we don't want our cells to be just a big puddle of liquid, basically. Okay, we don't want it to be super duper duper fluidic. We want to still sort of flatten it out a little bit. So what can happen here at the higher end, what cholesterol will actually do is help to fill in any spaces between these lipid bilayer. Between these fatty acid tails. So what's going to happen is, at a high temperature, there is a lot of energy, and that energy is causing the extra movement and energy to pull apart those fatty acid tails. Now, that's obviously disrupting a lot of these non-covalent interactions. So what cholesterol can therefore do is essentially wedge itself in between these fatty acid tails of these glycerophospholipids and hold them together. So it means that there are more of these non-covalent interactions that are occurring between cholesterol and these fatty acid tails, as opposed to if there wasn't any cholesterol in there, which means that it's going to decrease that fluidity because there's more things there holding those fatty acid tails together. Does that make sense, everyone? I hope that makes sense. So again, what is cholesterol doing here? It is decreasing the fluidity of the lipid bilayer, because essentially it's forming these attractions between these fatty acid tails. It's increasing the number of attractions that it would otherwise have. Okay, but that still doesn't answer what I brought up before over here. Sorry, that still doesn't answer the question. How can it both decrease fluidity and increase fluidity? I just discussed how it can decrease the fluidity. So let us now have a look at how cholesterol can then increase the fluidity. So what's going to happen here at, say, a very, very low temperature down here when the fluidity is going to be much lower, but we want it to be a bit higher here with cholesterol. What cholesterol will do is it will actually force the fatty acid tails apart. Sorry, I will go here. It's going to force these fatty acid tails apart, because if it's really, if the temperature is quite low and these tails aren't moving around very much and they're very close and compact together, what can then happen is that cholesterol can sort of wedge itself in between these fatty acid tails. Like, yes, it will have non-covalent interactions, these hydrophobic interactions between cholesterol and these fatty acid tails, but they won't be anywhere near as much as it would be if there was a very low temperature and these fatty acid tails were very compacted together. So what that essentially means is that cholesterol is acting as a spacer. It's spacing apart these fatty acid tails, increasing the fluidity of the lipid bilayer. So this is how cholesterol both increases and decreases the fluidity of the lipid bilayer. It increases the fluidity because it wedges itself in between these fatty acid tails. It also decreases the fluidity because even though it's wedging itself in between these fatty acid tails, it's still having these non-covalent interactions between fatty acid tails. So that is how we get this sort of more stabilized transition or change in membrane fluidity versus temperature. Now, to come back to this nice little summary that we did last week, but I've added a little bit to it. Now, when it comes to the degree of fluidity, or when it comes to what we were discussing yesterday, which was essentially, last week, which was the change in the melting point of a lipid, the longer the fatty acid tail, the lower that fluidity is. Or the longer that fatty acid tail, the higher the melting point is. Why? Well, for both of them, it's the exact same reason, okay? It's due to the increase in the non-covalent interactions that are occurring between these fatty acid tails. The second thing is looking at the degree of saturation. Now, the more unsaturated a fatty acid is, the melting point is going to decrease, okay? It's going to be probably a liquid at room temperature. When we're looking at fluidity, the presence of, or the increase in the unsaturation of a fatty acid is going to cause an increase in that fluidity. Because as we can see here, these kinks in the chain are going to cause disruption, and it's going to reduce the amount of non-covalent interactions that are going to occur between these fatty acid tails here. So these two things we did do last week, but the third and final thing I just wanted to add here is that cholesterol can increase or decrease the fluidity depending on the transitional temperature. So in other words, what the cholesterol is doing is it's broadening that transition range between sort of more rigid or more fluidic. Okay. Uh-oh. Oh, okay. We're back. Never mind. Sorry. So now, understanding that the degree of fluidity of a cell membrane, or of a lipid bilayer, I should say, rather, it comes down to the presence of cholesterol. It comes down to the degree of saturation or the length of that fatty acid carbon chain. Now, again, as I've made clear already, with our lipid bilayer, it's not the same lipid across the entire thing, okay? We have various compositions of different lipids. And what we can see here in our lipid bilayer is what is called a lipid raft. Now, when I say raft, as in like sort of like a plank of wood sort of thing that you imagine like people stranded out at sea would make, that's kind of what we're looking at here. What we are looking at here with our lipid rafts is that it's essentially a more dense region or section of lipids within the lipid bilayer. This will be a cluster of lipids that have, say, longer than average fatty acid tails, or more saturated, and it will be a lot more rigid and hold together. And what this can do is that it allows us to segregate proteins within the membrane. So we can then move things around, like how we've got this anchored protein or this caveolin here, sort of around together as a group. And that's kind of what I'm explaining down here. So this clustering of these proteins, what it does is, with the presence of caveolin, will accelerate this cell-cell signaling, especially when it comes to dimerization and facilitating endocytosis. So we can actually move that around and alter things based on the cell's need. Okay. Now, this asymmetry of membranes, especially when it comes to that lipid composition, this is what I was explaining before at the very beginning. And this distribution is very important because, again, structure equals function, and there are some things where it would just be pointless to have that type of, glycoprotein or something like that, or a lipid anchored protein on the inside of the cell versus the outside, because if it's used in, say, cell-cell signaling, or to communicate, say, apoptosis, so programmed cell death, we don't want that internally because it's wanting to communicate with, say, another cell. So that's just sort of yet another example there of this asymmetry of the lipid composition between different cells, just for different needs. Again, I wouldn't expect you to sort of memorize or be able to recite different examples like this. I just want you to be aware that there are there is an asymmetry there with respect to our membranes and the lipid composition. And again, this is following up on pretty much the exact same thing I was saying over here. So things like glycoproteins and glycolipids, they are always going to be on the outer layer of the cell membrane, just because of their role in cell recognition and cell-cell communication. Oh, excuse me. Let's jump up here. So some key points here, some sort of key summary things. Obviously, biological membranes are critical within cells and sort of facilitating life as we know it. These biological membranes are formed by lipids and proteins. And when we are looking at the fluid mosaic model, obviously the cell membranes aren't static and rigid structures. These are constantly moving around, primarily laterally. They can flip-flop. We can do that using an enzyme called flipase to help move things around. We also have these lipid rafts to help move various integral proteins around the cell where needed. And of course, they are asymmetric with respect to protein, respect to lipid composition for inner and outer layers, depending on the need and function of that cell. Okay, here we go. So let's have a look at the three main types of membranous proteins. There are three that we're going to be talking about. We're going to talk about peripheral proteins, integral proteins, and lipid-anchored proteins. Now, although we are going to be talking about all three, I want it to be made clear that peripheral and integral proteins will be my main focus here. Okay, the peripheral and integral proteins will be my primary focus. We will touch on lipid-anchored, but not as much. Now, the functions of these three protein types are so varied, as we can see down here. We see receptors, we see channels, gates, pumps, enzymes, all sorts of different things. So, let us now get started on peripheral proteins. Now, peripheral proteins are loosely associated with the polar head group of the cell membrane. This can be on the outside or this can be on the inside. And a key thing here, what's a really important takeaway message from this, is that it is loosely associated, okay? These are just non-covalent interactions that are holding the the peripheral protein to that the polar head of the lipid bilayer. And what we often find too is that it can actually move around depending on the function of that protein. So a perfect example of that is looking at the secondary messenger system, so of CAMP, which we're going to talk about in a moment. Now, what is also important here is that the peripheral proteins on a cell can be removed or disrupted by high concentration. Nice. By essentially changing the pH. Okay, if we change the pH, if we say put a urea solution on there, it can disrupt those non-covalent interactions, and that peripheral protein will fall off. Now, the next one is integral proteins. Now, an integral protein does not move side to side or just attach to the hydrophilic polar head. It will actually kind of pierce. It will go straight through that lipid bilayer. Now, what is also very, very important here with this integral protein is that it will contain stretches or segments of hydrophobic amino acids. And I'm going to talk about why that's the case in a moment. But what is also incredibly important here is just how bloody hard they are to remove and isolate. Because they are so buried in this lipid bilayer, the only way to isolate a integral protein is to disrupt it with detergent. It's basically to remove it by detergent to get rid of this lipid bilayer surrounding it so that you can isolate it. The problem with that is that detergent also denatures the protein, so it's very, very tricky to do. And indeed, it is why sort of peripheral proteins and globular proteins are much more well studied as opposed to integral proteins, because they posed a very significant challenge for a very long time in terms of isolating them. Okay, now, integral proteins, there are multiple sort of different types of integral proteins. I don't want you to worry too much about that. Again, we're not going to delve too much further into the different types of integral proteins. All I want you to just be aware of is that when we see an integral protein go through the lipid bilayer, it is predominantly an alpha helix. Okay, it is predominantly an alpha helix that comprises of the sort of intramembranous part of the integral protein. So here are just a couple of examples here in terms of integral proteins. So things like transporters, receptor ligands, gated ion channels, so like your voltage gated sodium and potassium gated channels that you saw with like action potentials in the heart and in skeletal muscle. We've got our immunoglobulin-like domains. We've got selectin, which we're going to talk about in a moment with our red blood cells. There's a wide different variety of integral proteins that obviously carry out a wide variety of roles. Now, I'm just going to very briefly just discuss the mechanism of action of water-soluble hormones here in respect to the secondary messenger system. So this is a good example here of what we're seeing with a integral protein and a peripheral protein. So hormones that do work in this system are things like, epinephrine, adrenocorticotropin hormone, follicle-stimulating hormone, luteinizing hormone, glucagon. Okay, so breaking down that our glycogen stores. All of these hormones, which I just realized you can't see, all of these hormones here are all water soluble. What this means is that they can't just move straight through the lipid bilayer and, mosey on their way through the cell to get to the nucleus and exert its effect. Excuse me. So what's going to happen here is the hormone, which can be any one of these are just an example, will bind to this integral protein, okay, this receptor. And what that is essentially going to do, this receptor is going to trigger GTP to bind to this G protein, this peripheral protein. And what it's going to do is once GTP is bound to it, it will kind of just shimmy. It'll kind of shimmy along the intracellular surface here on these hydrophilic polar heads until it reaches this enzyme called adenylate cyclase. Now, what adenylate cyclase does is it will take ATP, so adenosine triphosphate, and it will essentially cleave two of those phosphates off and convert ATP into cyclic AMP, so cyclic adenosine monophosphate. Now, what CAMP does is it will essentially act as a coenzyme and will activate our protein kinase. So for those of you who aren't aware what a protein kinase does, it is an enzyme that essentially phosphorylates various proteins or various enzymes. And especially intracellularly, you can think of like sort of a phosphate group as being like the on-off switch. If something is phosphorylated, it's turned on. If it becomes dephosphorylated, it's turned off. And what this will do is it will then activate our protein kinase here, which will then phosphorylate the next sort of downstream pathway enzyme and sort of away it goes, and with the pathway changing depending on the various sort of hormone that we're looking at here. So that was complicated. That was a lot of information. What was the point of that? The big thing is that we can see these integral and peripheral proteins working together in terms of cell-cell signaling or cell communication here, in this case being hormones, to trigger an intracellular response, which could be, production of proteins, opening of ion channels, opening of other, transporter proteins, all sorts of different things. Again, depending on what the cell or depending on what the body needs. Now, something I mentioned sort of 2 slides ago is that when we are looking at an integral protein, the section in which it passes through the lipid bilayer is going to be an alpha helix. Now, the big reason here... Never mind. The big reason here why integral proteins are predominantly alpha helix is because of this R group. Remembering the R group points outwards with an alpha helix. So if we can get those amino acids pointing outwards and they are hydrophobic, then what is going to happen is it's going to interact with those fatty acid tails and stabilize the alpha helix, stabilize this integral protein, making it very strong. Now, something to keep in the back of your mind, a good sort of rule of thumb, is that when we are looking at an integral protein here, and you look at the spacing between an alpha helix and the sort of average length or thickness, I guess you would say, of a lipid bilayer, it's often around 20. It's around 20 hydrophobic amino acid residues in a row are required to sort of create this alpha helical section of the integral protein. Okay, so that's actually surprisingly useful because that's something we can actually look out for. And this is something that we can use to be able to classify what type of integral protein we are working with. How do we do this? By a hydropathy plot. So what on earth is a hydropathy plot and how on earth does this thing even work? I'm glad you asked. What it does is we can look at the primary sequence of our protein, okay? So we look at just the order of amino acids that comprise this protein. And what we can do is then essentially categorize the polarity of each of those amino acids. So as I'm showing in this table over here, okay, our hydrophilic amino acids, they're going to have a negative transfer of free energy, okay? Whereas our hydrophobic ones are going to have a nice, real high positive transfer of energy. So what you can essentially do is that if you know the primary sequence of your protein, you can essentially look up each of those amino acids that are in your protein and then graph it compared to this free energy change. And what you're going to see here is that if it's positive, then it means it's hydrophobic, and if it's negative, it's going to be hydrophilic. Okay. So how does that then help us to identify whether this is going to be an integral protein? Because we know it's around 20. So we can look for a spike of around 20 amino acids in a row that are hydrophobic, and that is reasonable evidence on our hydropathy plot here to discern whether we have an integral protein or not, and if we do, sort of what type it would be. Okay, so here we have two examples that I've shown. So if we look at our hydropathy plot here, we can sort of see, big tank down here. This is super hydrophilic. A little bit of a spike. That's not really 20 residues in length, though. We look over here. Okay, we've got a decent sort of spike of hydrophobic residues, and there are approximately sort of 20 in a row here at the very least. But there's mainly one sort of cluster, I guess you would say here. And what that indicates is that with our integral protein, there's probably one alpha helical section that is passing through the lipid bilayer. And that is indeed correct. And this is the actual protein that we are looking at. Whereas if we have a look at our second protein down here, this one has way more clusters, okay? This one has 7 sort of spikes in total, which again clarifies or shows us over here that we have 7 different sections of our integral protein that are passing through the lipid bilayer. So this is what our hydropathy plot does. It allows us to discern and see how many times our integral protein will be forming an alpha helix and sort of passing through that lipid bilayer. This is just sort of a very sort of quick side note here, just with looking at beta barrels and transmembrane channels. Alpha helix are obviously much more common. It's not exclusive. What we can see here is the formation of beta barrels, which can be very useful for forming a pore. I'm not going to be discussing beta barrels and looking at transmembrane channels like this in any reasonable depth. I just simply wanted to bring this to your attention that beta barrels within the cell membrane as a pore or a protein channel definitely do exist. Okay, what we'll do, guys, let's have a quick 10-minute break there. Stand up, stretch your legs, grab a cup of coffee or something, and we will continue on in about 10 minutes. Ooh, there we go. Hopefully that's all working well. Excellent. Okay, let's do this one. There we go. So we've spoken about our peripheral proteins. We've spoken about our integral proteins. The last one I want to talk about quite quickly is looking at lipid anchors. Now, what a lipid anchor is, it is essentially a protein of sorts that will attach itself covalently to the lipid, okay, making it incredibly strong and very, very rigid. What this means is that if you want to try and cleave or get rid of that protein, things like using acids or using salts, stuff like that, just completely ineffective. It doesn't do anything. So what's going to then happen is that if we join our protein to a lipid, and we get that lipid sort of anchored into the lipid membrane like we can see here, it just makes it a lot more structurally supported and a lot more rigid there. That doesn't mean that we have no control over it, however. We can use an enzyme called phospholipidase C, and it will essentially just cleave off that protein as needed. Okay, what we're going to do now is sort of wrap up membranous proteins by looking at red blood cells. Now, red blood cells, in terms of sort of the cellular structure, are one of the simplest that we do see in mammalian cells, mainly due to its function, and that it just possesses far fewer cell membranes because of just it doesn't really need them. Now, if we do an SDS page gel and we have a look at some of the main protein bands, there are around 15. However, there are only really three of these proteins that are just super important, okay? The alpha spectrin, beta spectrin, and glycoporin make up sort of around 60% of the total membranous proteins that we do see within red blood cells. So the first one we're going to quickly talk about here is glycoporin. Okay, so this is a single transmembrane integral protein that we were looking at before with our hydropathy plot. And quite interestingly, this was the first membrane protein in which the amino acid sequence, so the primary sequence here, was completely determined, which is pretty cool. Now, what glycoporin... its sort of primary function here is that it's incredibly hydrophilic on the outer part here. And what this does is it sort of acts like kind of like a buffer, I guess you would say. Like this is still a current area of research, but the main function of it is that it works like the rubber lining of a bumper car. Okay, it sort of stops the cell from getting damaged and not sort of sticking to or rubbing against other cells and getting damaged in that way. It sort of helps to buffer the cell around. Also, quite interestingly, this is the one of the proteins that the malaria parasite exploits or takes advantage of in order to gain entry into our red blood cells. The next one here is spectrin, okay? Spectrin here is a type of sort of cytoskeletal protein. And what it does is that it allows the red blood cell to be flexible, but still somewhat rigid. Now, how I want you to think of this is almost like bed springs, I guess, okay? Like springs in your bed. It allows your bed to be, or the mattress, I should say rather, to be more rigid, but it's not like sort of sleeping on the floor. It can still be flexible. It can still sort of change its shape when you lie down to go to sleep, but it's not going to just completely collapse on you, that sort of thing. Or you can think of it as like a trampoline, okay? Like the fibers on a trampoline. They will stretch, but they are still able to return to their original shape once the stress has left. And the last one I very want to briefly talk about is ankyrin. Now, what ankyrin does, again, if we say return to my little trampoline example, if this is the trampoline mat that we sort of jump on, we need something to hold it together. And that's what our ankyrin is. It's kind of like a staple. So if we have a look at this image here, which is sort of representing the innermost part of the plasma membrane for a red blood cell, what we can see is that we have these spectrin fibers, and they are held in place. They are anchored in by our ankyrin here, and using sort of actin molecules to sort of hold it in place. So that way, if say we have our red blood cells trying to get into, the really sort of tight nooks and crannies of our capillaries here, they're not just going to get clogged and get stuck. The red blood cell will able to sort of be compressed and change its conformation, and then kind of, to sort of squeeze through, and then will spring back to its original shape without any damage to the red blood cell. And that's it, guys. That is it for plasma membranes. Nice and straightforward, because we did a big brunt of the work last week. So what we're going to do, guys, is we are going to now transition to our Tute workshop. So, I'm not going to say let's take a 10-minute break, because we kind of just had one. So I'll give you guys 5 minutes to get Zoom loaded up and move over there. And yeah, other than that, I will see you all in the Tute workshop. See you soon.
Pretend I'm fine, but I know you can hear me sleep talking at night. do